Medicos, in this video I am going to explain genetic disorders. Now we know that as the name depicts that disorders that are related to our DNA. Like 50% of early abortions are due to DNA abnormalities. So genetic disorders are very common. Now let's classify the genetic disorders. We have first classification of disorders that are related to mutation of a single gene. Like whenever there is a mutation of a single gene, there will be a genetic disorder, and if that gene follow the Mendel's law, then they are called as Mendelian disorders, and they are penetrant. What is meant by word penetrant? Like presence of mutation is associated with disease in large number of population. Now the second classification is chromosomal disorders. Now we know there can be structural as well as numerical disorder that can lead to the chromosomal disorder, and again it has a high penetrance value. Third thing is multigenic disorder. Like this is a complex multigenic disorder. These are the complex genetic disorders which are influenced by multiple genes as well as environment. Like we can say hypertension, atherosclerotic diseases, like they are multifactorial disorders. They are influenced by multiple genes as well as their environment. Like hypertension is associated with walk, with weight reduction, etc. Now the fourth thing is single gene mutation, but it is not known as Mendelian disorder. Why? Because it doesn't follow Mendel's pattern of inheritance. Here, the first that was the disorder related to mutation of single gene was following the Mendel's pattern of inheritance. That's why we call it as Mendelian disorder. Now, a fourth category of single gene mutation is also present, but this doesn't follow the classic pattern of inheritance. So basically, these are the four types of genetic disorders reasons. Now, I'm just going through their introduction, like we have to study it in detail later on. Now we are going to study mutation. What is meant by the word mutation? Mutations mean any permanent change in DNA, and now this mutation can be of different types. Like there can be parent mutations, there can be deletion, insertion, there can be trinucleotide repeat mutation. Now we are going into the details of these points. First of all, there can be point mutation. What is meant by point mutation? That there can be insertion of some different base, so it is going to alter the code of triplet of bases, and that is going to encode a different amino acid. This is called as missense mutation. And if that inserted base results in some stop codon and there is low transcription of that protein, there is like less volume of that protein, that will be called as nonsense mutation. Now in missense mutation there is different amino acid, but in nonsense mutation there is expression of some stop codon which can lead to the early termination of protein transcription. Now this missense mutation can be of two types, conservative and non-conservative. If the inserted base is going to result an amino acid that is biochemically similar to the original one, like it will not cause any change in the function of protein, then this is called as conservative missense. Like there was mutation, but the mutation results in some amino acid that was functionally that was biochemically alike to the original one, so there is no there is less change in function of that protein. That is called as conservative missense mutation. And non-conservative missense mutations means if that substituted base results in an amino acid that is not biochemically similar to the original one, so there will be no more functioning of that protein that was supposed to do. So this is called as non-conservative missense mutation. This was all about the point mutation, and this point mutation was in the coding sequence. Now what happens when there can be point mutation in non-coding sequence? We know in our non-coding sequence there are introns, there are promoter enhancer sequences. Now if the mutation happens in this promoter or enhancer sequence, now what's going to happen? It can interfere with the binding of transcription factor, and there can be marked reduction and total lack of transcription. So what happens when a point mutation occurs in promoter or enhancer sequence? There can be marked reduction or total lack of transcription, like in thalassemias. And if the point mutation occurs in intron portion, then it is going to affect our splicing. And if there is defective splicing, then there is failure to form mature messenger RNA and then protein. This is how point mutation in non-coding region are deleterious as well as in the point mutations in the coding sequence. Now the third thing is deletions and insertions. Now consider an example. If there are true triplet code, this is a DNA sequence like C A G D A. Okay. If the number of bases inserted or deleted are is equal to 3, like consider there is deletion of these 3, then or there can be insertion of the 3 bases that is going to transcript an other protein, that is going to code an other amino acid. Like there can be increase in number of amino acid or there can be decrease in number of amino acid depending upon the number of triplet added in our sequence. And this is going to, there will be no frame shift mutation, like we can read the frame exactly, and there will be only this one will encode another extra amino acid in our proteins, and our proteins will be non-functional. Now if there are the number of bases inserted or deleted are less than 3, consider if there are 2, there are 1, like if I am going to remove this, now there is frame shift mutation. I am going to study it like this: CAG TCTA ATT. Like there is thorough frame shift mutation. There are abnormal amino acid now that will be encoded by such sequence if there is insertion or deletion of any less than 3 number of bases. This is how there is frame shift mutation. I have given here an example, like there are 1 2 3 and 4. If I am going to remove this, then whole the sequence, whole the reading frame will be different now. The fourth thing comes here is trinucleotide repeat mutation. Here is amplification of sequence of any 3 nucleotides. Like a triplet will be amplified, like the sequence of a 3 nucleotide will undergo amplification again and again, and there will be large amount of that protein, and there are a lot of examples under this mutation. There can be Fragile X syndrome, there can be Huntington disease. In Fragile X syndrome there is CGG, tandem repeats of CGG. Like in normal person there are usually 29 repeats of this sequence, but what happens in Fragile X syndrome, there are 250 to 4000 tandem repeats of CGG, which are going to cause a situation which is known as familial mental retardation 1. This happens in trinucleotide repeat mutation. This was the overview of genetic disorder. Now in my next video I will explain each disorder in detail one by one. Thank you for watching. Bye bye.